you'll be talking about Ampliclust.
Thanks for having me. And I'm Xiyu Peng. I'm a second year PhD student from Iowa State University. Today, I'd like to talk about Ampliclust. It's our model-based approach for denoising the Illumina Amplicon data. And at the beginning, I'd like to have a very brief introduction about the problem we are focused. Amplicon sequencing actually is a very powerful tool to understand the microbiome community. People often sequencing the 16S RNA to identifying the components in the community. And the whole analysis usually begins with the construction of OTU. And here is a graph I modified from Data2's paper and show the whole workflow. The left is actually the real sequences in the sample. It's our goal that we want to find. However, in the middle is the data set we usually get. It's Amplicon reads, and it's full of the errors introduced by PCR and sequencing process. So it's really hard to identify which are real, which are errors. The previous strategy is to classify those reads into 97 similarities into OTUs. And then the downstream analysis based on the construction of OTUs. However, the OTU has very low resolution. In this example, they actually have the 4 species in the sample. However, we only constructed 2 OTUs. And this may lead some problems in the downstream analysis. So recently, new methods are developed to focusing to find the exact sequence in the sample. This is also the objective of our algorithm. We want to infer the identity and relative abundance of error-free biological sequence in the sample. And we propose a mixture model for this problem. And here, I like to present my model as in the form of likelihood function. Suppose given the data R and Q. R is the nucleotide sequence, and Q is the coordinate score sequence. And I suppose that each read in this data set is IID generated from this mixture distribution I showed in the red box. And this mixture distribution have K components and denotes by the abundance, relative abundance pi. And this part is denotes the probabilities of the reads it generated from a specific haplotypes K. And I want to answer the question, estimate all the parameters in the equations, so maximize the likelihood function. And however, this model do not answer the question that how many haplotypes in the samples. And so, I treat these problems as a model selection problem. And we use the main idea is to use the Bayesian information criteria to select a better model in order to determine how many components in this mixture distribution. Actually, we face a lot of problems when dealing with this problem. The first is the over-parameterized on the haplotypes. And how we deal with this problem, we use the Drux counter model. Actually, it's a classical evolutionary model to model the relationship between the haplotypes. Then comes another problem that is it's very hard to deal with the hierarchical model, and I do not want to use the Bayes method. So that actually I calculated BIC will likelihood approximately maximize. Since it's not the original BIC, so I called it the modified BIC. However, there's still a lot of problem for the mixture model, and it's really easy to get into a local optimal. Okay, the model is perfect. If we give a very good start point, we will have the amazing results. However, it's really hard to find that start point. So we are inspired by Dippler, and we designed Ampliclust. Actually, Ampliclust is designed as the initiator of the Ampliclust, but later we found its high accuracy and efficiency, and it already have the better performance than other algorithms. And actually, this Ampliclust is designed based on the Ampliclust model, and we add a lot of assumptions here to make the algorithm more efficient. So it's actually the simplified version. So the main idea of the Ampliclust is very simple. I try to present in a very simple way. And beginning, I will want to compress the data set into the groups of the unique sequence. And the blue circle denotes the true abundance of each unique sequence. And however, the green circle is the number of the sequence that we observed in the data set. and they have overlap, but it's not exactly the same. So the main goal is we want to infer the blue circle from the green circle. And if we made assumption that the error rate is constant over the whole sample set, so the overlap between the two circles should be a constant proportion of the blue circle. So we build a model to infer the overlap between these two circles. Now here is our model. And I like to call the overlap is actually the scaled true abundance of the unique sequence, and it should be equal to the observed abundance of unique sequence and minus the number of reads comes from the other haplotypes. And here, and specifically, I like to I use every read in this unique sequence sample and calculate the probability of each read that is generated from a specific haplotype K. Actually, I like to highlight this probability here. I use base rooms here. It's actually the posterior probability that this read is generated from a specific group. I do not only consider the sequence identity and also quality information here, and I also consider the abundance of the haplotypes and also the unique sequence. So that's why our algorithm works. So this is a workflow how we select the haplotypes. At the first beginning, we'll construct our unique sequence table from the highest to the lowest, and then we select haplotypes step by step based on the relative true abundance we calculated before. And we also evaluate each newly selected haplotypes based on our modified BIC. And then if the BIC support a bad model, and we will we'll think this is a real haplotype, and we're based on the current model, we'll update our relative true abundance, and then we'll continue this circle until we reach the lower bound on the relative true abundance, and we do the read assignment based on our likelihood model. We simulate our data set based on model that is very similar to ART, that we actually learn the quality profile from the real mock data, and then we treat the quality score as literal. Here, actually, then we compare our algorithm with 3 very popular algorithms based on the testing data set. Well, the graph in the left is actually the performance of the algorithm on the 2 different data sets. Maybe you cannot see very clearly. And the left is performance on the even abundance data set, and the right is a performance on the uneven abundance data sets. And actually, our result is shown in the red bar here, and the y-axis here is actually the proportion of the recovered haplotypes. And the graph in the red actually shows the performance on data sets that's with the increasing similarity of the haplotypes. And our result is also the red line. And the 2 graphs show that our algorithm actually have it's the most robust among all the data sets compared to all the other algorithms. And also like to evaluate the performance on clustering. Now, currently, I use the adjusted random index. It's actually the measurement of the similarity of the 2 clusterings. In each line, I highlight the best performance on the data set, and I found our algorithm actually have the best performance on most of the data sets, especially for data sets that have the very high similarities. Okay, and then we also test our algorithms on the mock data set, and we selected mock data set from Data Tools paper. It contains about 2 million forward reads, and also it's extremely unbalanced. This is actually the most difficult data set to be classified. No algorithm could do perfectly. And I like to claim that our work is still in progress. We haven't dealt with the in-depth errors, and we still have not found a way to get a very accurate error profile, since our algorithm is very sensitive to the error profile. So I don't want to talk about the false positives. So let's just focus on the first 9, and it's actually the number of the sequence that could 100% match the reference. And our algorithm actually can find more haplotypes than 103 and deeper. And data 2, I actually find more, but the 4 more haplotypes that data 2 found is below our threshold. So, and in the last, I'd like to highlight our algorithm is actually a probabilistic model-based approach, and it just has resolution and accuracy, especially for the data set that have more similar haplotypes. And I also implement the whole algorithm in C. And currently, I think time is acceptable for the mock data set. It spent several hours, and we still try our best to accelerate it. And our future work is firstly to do the error estimation of the Amplicon. Since actually Ampliclust did error estimation, but Amplicon is a simplified version, so we just input the error profile. And we also need to model the in-there errors.
Thank you very much.
Yeah, okay.
Thank you for your presentation. So thank you everybody for joining this session today, and I would like to thank all the speakers of today and for speaking, and whoever wants to join us for dinner, we're meeting at 6:30 downstairs at the bar in front of the main entrance, and we're trying to find a restaurant for dinner, so you're very welcome to join, and we hope to see you tomorrow also in our next